Thank you, Lou. So I am very excited to announce the next set of speakers. We're going to wrap up the day on a pretty fun note, maybe get our head above the infrastructure and learn a little bit about the discoveries that are happening on top of Openstack. Many of you probably know that Openstack has a very rich history in research, of course, starting out with NASA, and you've probably heard that CERN is running at a very large scale. But a year ago, when we were in Barcelona, we heard from Dr. Rosie Bolton about the Square Kilometer Array and what they're doing to build that out using Openstack. And 6 months ago in Boston, we heard from Dr. DANIELA, who's one of the leading roboticists at MIT, about the research that she's doing powered by Openstack. So today we're going to hear about a local story, the Nectar Research Cloud, which is powered by Openstack, and it's actually run collaboratively by several different organizations. So I'm going to bring out two of the folks who are helping to run that cloud, which is Dr. Stephen MANOS from the University of Melbourne and Dr. Steve Cunett from Monash University.
Thanks, Lauren.
Thank you, Lauren. Hi, everyone.
Hi, everyone. We're here to celebrate 6 years of sustained successful delivery by the Nectar program. It started way back in 2011, where as a national community, we got together and asked the question, what is the right infrastructure that would enable researchers to work together? The ambition was to support the connected researcher, where they would be connected to other researchers, to communities, to industry, to advanced analysis, and to vast amounts of data. And yes, you can guess it, the answer was Openstack.
So since then, Openstack has underpinned research outcomes of hundreds of research groups across Australia and also supported the collaborative endeavours of thousands of researchers across Australia and beyond. And as a national community of Openstack operators spread across many cities and states, we have also contributed to the growth of Openstack internationally. For example, we were an early adopter of cells since the Essex release years ago now, where Nectar-funded stackers worked closely with Rackspace and others in early deployments. This was critical to achieving a single cloud that operated across 7 organisations and 5 states in Australia. And as of a few months ago, we've had New Zealand join us as an international cell of the Nectar Research Cloud. We've also learned a lot about distributed deployment, lessons of which have been proliferated through Openstack user committee forums, such as the large deployments team.
The same paradigm of global open source innovation has occurred higher up the stack. Researchers create their own bespoke environments. We call these virtual laboratories. And to aid that cause, Nectar-funded stackers participate in the Scientific Special Interest Group. They focus on the virtualization of specialized hardware, such as GPUs and interconnects. At times, what this means is at times, the researcher programs the computer that they actually need. They design it.
And what Stephen has described is really the essence of open source infrastructure. We believe that open computing, open platforms, open communities and open APIs are a critical foundation of research. APIs will become and will be the universal language of discovery. Now onto our 2 great science talks. These 2 talks you're about to hear do exemplify the goals of collaboration and the integrative nature of Openstack. And they also demo some critical research which is going to impact everybody in this room. First up, I'd like to welcome to the stage our Professor Brendan Mackey. He's the Director of the Centre for Climate Change at Griffith University, and I'd like to welcome Brendan to the stage. Thanks, everyone.
Thank you.
Thanks, Steve. Thanks. Clicker. Well, good morning. It's a pleasure to be here. I've been asked to talk to you about our biodiversity and climate change virtual laboratory, which is really helping researchers to address one of the big challenges we have today, our biodiversity, all the species that live on Earth, the plants and the animals, the wildlife, also the things that want to bite you and kill you, like vector-borne diseases and viruses, and how they're being affected or may be affected by human-forced climate change. And all of this requires a lot of data and compute, analytical capacity. And by a lot of data, I mean a lot of scientific data. These are deeply curated data sets that conventionally have been very much siloed, that need to be brought together. The first are the biological observations that dedicated field researchers gather when they go on remote field trips to places like the Amazon or the Congo. The second are the data we have about environmental conditions. A lot of this comes from satellites and from instruments, for example, bobbing around in the sea. And the third are the forecasts we have, the GIS-based forecasts we have of future climate. And there's not just one future climate, there's hundreds of future climates, because there's over 40 global climate change models, many different scenarios, and many different time steps and parameters. So we need to be able to, researchers need to be able to bring all these data together to look at what impact these might have on our wildlife species. So for this reason, we work with Nectar and a team of collaborators around the country and internationally to develop the Biodiversity and Climate Change Virtual Laboratory. And we had 4 aims. One was to remove the technical barriers to accessing biodiversity and climate change modelling. This is not because our researchers are stupid. They're actually very clever. They're very good at mathematics. They quite like writing R code. But there are a lot of barriers to bringing data from these various silos together that I was referring to. We want to raise the standard of species distribution modelling, because we need very robust models in order for the information to be taken seriously and used in the real world. We wanted to simplify the access and visualization of these disparate data sets and provide training, especially for early career researchers. And I'm going to give you a case study of 2 species, 2 Australian species, leadbeater's possum, arguably one of the cutest animals on Earth, big baby eyes, and its habitat, the mountain ash, Eucalyptus regnans, which, as you can see from the person there, is actually the world's tallest flowering plant. And there are 2 big threats to the little leadbeater's existence. One is a lot of people are trying to kill it by turning its tree into a cardboard box, right? The other threat is climate change. How is the climate change going to affect that animal's habitat? So I think this is where we go to a video of the research users interface of the VL. So the VL user interface enables the user, the researcher, to quickly search and find the climatic environmental data that it needs for the modelling, to find and discover the biological data. This might come from a repository in Australia, it might come from a repository in Europe, a herbarium there, for example. Researchers can upload their own data if they have been doing fieldwork in the Amazon or the Congo and have some field data of their own, which is uploading now very quickly. And then you can run different kinds of experiments. Here we're looking at a suite of different kinds of statistical distribution modelling experiments using advanced regression techniques. And when ready, the experiment can be run. You get all the kind of visualizations of the normal diagnostics you get, for example, in this case, from a regression model. And of course, you can get the R script that's generated by the interface if you so wish, and you can look at the results and compare multi-model runs, etc. You can then take that model of current climate and project it into the future. We have access, as I said, to many, many different future climate projections. And here's what we end up with after we do that little exercise. On the top is the current predicted distribution as a function of climate for mountain ash, the home of the mountain pygmy possum. At the bottom is where the pygmy possum current distribution is. And then in the middle where they will be by 2075, and then over on the right-hand side, the difference between the two. And this is what we're looking at. This is southeast Australia, which is just south-west of where we are in Sydney, down towards Melbourne Way. The red is mountain ash, the blue is leadbeater's possum, and the black is where they overlap, which is, of course, where the animal can actually exist. And what the modelling is telling us is that by 2075, the area of overlap shrinks. So unfortunately, climate change is not a good news story for Leadbeater's possum. So how does this all work? Well, the user, of course, doesn't see anything that's in the middle. That's the system. On the right-hand side is the external data. So the user is importing or exporting data from our external data holders through a web API. Is designing the experiment in the system, creates a job file. It gets sent to the virtual machine. Next has a cloud of about 30,000 cores. The data is fetched, the script is run, the output is created. So using web and Swift APIs, the data information is transferred from the user to the system to the external data and back again. There's always a backup in the Nexta cloud, and there's short-term job files held in the BCC local database. And this is an iterative process. You don't want to run the experiment once, you want to run it multiple times in order to get a refined model, and eventually you want to share that with the world. So all that the user sees is the left-hand column. It's like a very sophisticated app, if you like. And the good news is, before I finish, that you don't have to be a researcher. Anyone here in this room or anyone anywhere in the world can go to bccvl.org, register, and explore how your favourite species might change as a function of future climate. Thank you.
Thank you, Brendan, for that brilliant example of how collaborative infrastructure has enabled researchers to work together. You might be able to sense as well that that same collaborative idea is moving towards social computing. Up next, our example will show you how Openstack and high-performance computing is coming together. Gary Egan is a director of Monash Biomedical Imaging, and he's up next. Thanks.
Ancient Egyptians sucked it out of the noses of their dead and then discarded it. Aristotle thought it was a radiator for cooling the blood. But by the end of the Middle Ages, the brain had won respect. No longer spurned as mere cranial stuffing, the brain was recognised as the seat of cognition, key to bodily function, a thing of intrigue, even beauty. In the 18th and 19th century, findings came thick and fast. Electricity's role in nerve function was spotted by Luigi Galvani, inspiring in a roundabout way Mary Shelley's Frankenstein. Louis Victor Le Bonnier helped show that different parts of the brain had different functions when he lost his ability to speak, but none of his other faculties, enabling Paul Broca to map brain areas that made speech possible. Santiago Ramon y Cajal showed that each spider-like brain cell was separate, not fused to its neighbour. That was key to understanding plasticity, and therefore the brain's ability to learn, remember, and recover. Come the 20th century, another patient, Henry Molaison, taught us about memory by losing his own on the operating table. The removal of parts of his brain to treat seizures left Molaison unable to remember new events, but still able to learn new physical skills, and allowed Brenda Milner to deduce that there are different types of memory located in different parts of the brain.
G'day. My name's Gary Egan. I'm the Director of the Australian Research Council Centre of Excellence for Integrative Brain Function, and I'd like to share with you today some of the research that we're doing using the Openstack tools. Nearly 50 years ago, researchers started building the first brain imaging scanners, and you see here a couple of photos from back in the early 70s. The challenge then was to just look inside the living human brain. About 30 years ago, the first images were taken of seeing people do mental tasks, seeing things, hearing things, thinking about things and doing work. And so our challenge has been to map both the structure of the human brain and the functions, and relate the functions to the microscopic detail of the brain that was first discovered by Ramon Cajal. So this challenge is of great interest, and we also look at outliers like Homer Simpson, and we're very interested in the variability across all of us in how our different brains work and function.
Today, we have brain imaging technologies capable of eavesdropping on the ebbs and flows of brain activity from second to second, over distances just the breadth of a few human hairs, without harming a single brain cell. There are techniques like optogenetics that use light to control living brain cells, and transcranial magnetic stimulation that can speak to the brain in its own special language, rousing cells to fire or to rest, to meld into fresh circuits or go their separate ways. And all of this is coupled to insanely powerful computers capable of crunching the torrents of new brain data. International teams of scientists are jumping at the opportunity to use these tools to find out more about the brain. At the Human Connectome Project, they are mapping the brain's connections and relating those wiring diagrams back to each person's cognitive abilities, personality and genetics. At the European Union's Human Brain Project, they are simulating the brain in a computer, arguably the only way to understand such a complex beast. At the US Brain Initiative, they are developing novel neurotechnologies to explore how brain cells and circuits interact in time and space. And at the Australian Research Council Centre of Excellence for Integrative Brain Function, we are working out how the brain interacts with the world, how it shifts attention to the things that matter, predicts what might happen next, and makes decisions, continuously, subconsciously, and at breakneck speed.
The Centre of Excellence is based at Monash University in Melbourne, and there at that facility we have now a wide range of brain imaging technologies, as you can see in this slide, measuring the microstructural connectivity, as well as maps of function, and most recently, metabolism. We also use the massive supercomputing system there at Monash to both process these very large data sets and also to visualize the incredible complexity of the brain in 3D immersive reality environments. So today, we're able to map dynamically brain function, as you see in this video, which is data from a real experiment we conducted a few years ago, where firstly we mapped the structure, as you see in this case, with different sections through the brain. We then ask individuals to do tasks inside the scanner, and we can see the flush of neural activity that occurs at the onset of the task. Throughout the task, there's continued activity, and then at the end, there's another flush when you change your motor activity. So this is incredibly exciting for us. It's enormously great fun, and looking to the future, we actually now have the next generation of brain scanners which are becoming available. And as is the case for the square kilometre array and the large telescopes, as we peer further and further into the brain, we need to increase the resolution of our scanners. So recently there's been the development of the next generation of ultra-high field MRI scanners, and you see in this little cine loop some of the brain sections through the back of the head. This is actually the occipital lobe where the visual processing occurs, and underneath it the cerebellum. And we can now almost nearly see individual neurons in living people's brains. So that's phenomenally exciting. Monash University is located in Melbourne, and the Australian synchrotron is also located at the Monash campus. And I wanted to finally share with you some of the incredible, exciting data from the synchrotron beamlines. This facility uses the characterization virtual laboratory, which is built on the Nectar stack, and our research work, particularly using imaging, would be impossible without that underlying computing infrastructure. So here's an image of a rodent. It's not as pretty an animal as Brendan's that we heard about earlier. It's a rat. And in this research, we're using synchrotron phase contrast X-ray imaging to be able to look not just at the whole animal, but in detail inside the animal. And this is an amazing new technique for 3D volumetric ultra-high resolution CT scanning. So we're looking at structure here in this video, but in fact, when you do it very fast with the synchrotron's high flux of X-rays, you can actually infer function by looking at dynamic structural changes, as in the case of respiration in the lungs.
Brain science is entering a new era that will transform the ways we learn, play and work, even our very sense of what it means to be human. To share that journey of discovery, to contemplate its ethical and social issues, and to co-create the future together, the Centre for Integrative Brain Function runs the Brain Dialogue, a program of events, online discussions, citizen juries and participatory science. Visit us at www.cibf.edu.au.
So thanks to Openstack, and thank you for your attention.
Thank you very much to these researchers who took time out of their busy schedule to come talk to us. We really appreciate it. We also want to give a shout out to the Science Working Group who just updated their book about HPC. So a big shout out to the Science Working Group. They'll be meeting this week. There'll be quite a few different sessions. If you're interested in hearing more about this research that's happening, you can also check out the University and Research track on the schedule.
Last but not least, please do not forget to have a good time with Openstack. Thank you.